When this started happening, it was very clear to me that even though it's not a basic research question, this is the most important thing that I could do at this time. Over the past 8 years, one of the major focuses in our research group has been on learning how to design antibodies on the computer. And we've shown preliminary data that we can do this in atomic accuracy. We can actually design antibodies on the computer, then go to the lab and experimentally verify that they have the shape that we designed and actually have some of the functions that we designed. Antibodies are essentially our body's most important lines of defense against viruses. In our immune system, there are specialized cells called B cells, whose sole purpose is to develop antibodies to fight off infection and then to mass produce these antibodies. These cells become memory cells. And then the next time we face the same virus, we're essentially immune to that virus. This coronavirus is unique and new. Our bodies have never been exposed to this particular virus. And so when we face this new one, our antibodies are simply no good. In my lab, we had already started working on SARS, the previous pandemic. And so when this started, we were actually quite ready to switch over to coronavirus because the viruses are quite similar to one another. So we already knew what the vulnerabilities are of coronavirus. And so we're targeting these vulnerabilities with our tools. What we're doing on the computer is essentially docking all of the antibodies that we can lay our hands on against these vulnerabilities, and then we're refining the antibodies. Instead of relying on the immune system, we're refining the antibodies to bind more tightly. We know from SARS virus again that binding to these vulnerabilities can block infection. And in this process, we're producing millions and millions of different antibodies, and then we go to the lab and actually create, in a test tube, all of these millions of antibodies until we find a hit that's validated, and then we can provide it to our collaborators who will verify that the antibody is not just a good binder, but that it can actually block infection. We're trying to do whatever we can in the process of developing a new vaccine or even in treatment against this deadly virus. It's widely understood that this is a unique time. People are sharing both reagents, probes for experiments, their information, data. We got the molecular structure of the coronavirus spike protein before publication from our collaborator, just as a, it's not just goodwill. Can you do anything with it? He's also sending us material so that we can do the experiments in our lab. Once we're done with this, other people will pick our proteins up and verify that they are actually neutralizing. So this requires a massive interdisciplinary effort, and we're getting huge support from both within the institute and from outside. Everybody is trying to help, even though people are shutting down in some senses. Whenever you say this is a corona-related research, they say, okay, we'll prioritize this, and then they respond immediately. It's really a unique time and a very, I'd say, inspiring time for me as a scientist to see how the entire scientific community is coming together to solve what is probably the most important problem of our age, and where science can actually have such an immense impact on human society.